Welcome back. This is Chapter 17, Part B on the cytoskeleton, namely actin filaments. The chapter is quite nicely organized into the structure and then the function of actin filaments pertaining to cells, namely animal cells. And the last part of the chapter looks at how striated muscle, skeletal muscle, works with respect to contraction. The primary role of actin filaments in animal cells is to maintain shape, but as we'll see a little later, they can also be involved in motility and also in mechanical events. The use of actin filaments has been propagated in animal cells to the greatest degree. They are also present in other cells, but animal cells have done this to the benefit of themselves. In Panel A, we can see the existence of microvilli, which increase the surface area of a cell, and those microvilli are maintained, their shape is maintained by the presence of stable actin filaments. In Panel B, we see some contractile bundles in the cytoplasm of that cell, which allow that cell to maintain its shape. In Panel C, we see a cell that is moving along the substratum towards the bottom right-hand side. This is what you see when you look at an amoeba cell crawling along the substrate. And then finally, in Panel D, we see what happens during cell division. As the two daughter cells emerge from the mother cell, we get this belt of a contractile ring that appears and constricts until the two cells pinch off. We'll study Panel D in more detail in the next chapter. Actin filaments are made of globular proteins. The proteins come together as monomers, and the monomer exists in the shape indicated here. The monomers form a filament as shown in Panel B, and the filament has polarity. So because of the existence of different amino acids in the monomer, one end of the filament is called the minus end, and the other end of the filament is called the plus end. And the importance of that will become clearer in the next few slides. But these filaments are normally about 5 nanometers in thickness. So they are normally the smallest in terms of diameter of all the cytoskeleton elements that exist. Being thinner is not necessarily a bad thing because it allows these filaments to be more flexible than the other elements that we discussed in the previous video. But they do tend to be shorter in length, the average length being about 100 nanometers. The monomers do have the ability to bind ATP and ADP, depending on the circumstances that exist in their environment. That's going to become important also in subsequent slides. A fascinating observation made by scientists with regard to actin filaments is that they do grow on the plus side. So the plus side of the filament will polymerize, whereas on the minus side, the removal of the monomers takes place. So one end is growing, the plus end, whereas the minus end is losing monomers and is shrinking. That leads to something called treadmilling. So we can imagine if the rate of addition at the plus side balances the rate of depolymerization at the minus side, even though the length of the polymer stays the same, there is a treadmilling effect where the polymer is growing at one side and diminishing at the other, but still staying the same dimensions. The treadmilling effect uses ATP to build the plus side, and the hydrolyzation of the ATP at the minus side results in depolymerization. Thus, the plus side is associated with ATP, and the minus side is associated with the ADP. So this is important to remember. So now that we know what treadmilling is, let's compare that with dynamic instability that we saw with microtubules. With the phenomenon of treadmilling, there is a flow of subunits from, in this case, the right-hand side to the left-hand side. So the subunit numbered 1 is just being added right here. But a fraction of a second later, it is now moved to this position, and then to this position, as new subunits are added to the plus side. At the same time, subunits are being removed from the minus side. So this is like a treadmill where the platform is moving, but the treadmill doesn't change dimensions. So this is what actin filaments do. In contrast, dynamic instability is where the subunits are added and removed at a high rate from the same end. So here, this will be unit number 1, and this would still be unit number 1, as well as here, here, and here. But this unit here that's been added recently will be right here, and new units will be added until such time as the shrinkage, and those units will be removed, including the one that we were interested in right here, until a point is reached where it starts growing again. So this is a dynamic process with this wholesale expansion and contraction, whereas in treadmilling, everything takes place at the appropriate side without the other side being involved. This slide here shows us that there are many opportunities for actin filaments to be utilized in many different functions. But polymerization is necessary, and association with accessory proteins is needed by that cell. So in different cells, the presence of different concentrations of different proteins will result in different functionalities. As we'll see, in muscle cells, we can have the actin myosin filaments produce muscle movement. In other regions of other cells, we can have cross-linking of the actin filaments to produce a matrix, three-dimensional matrix, near the cell cortex. That's the inner surface of the plasma membrane. We also have severing proteins. These are like enzymes that cut the actin filaments at their longitudinal length, as well as capping proteins which prevent the further elongation of the positive end of the actin fiber. And also we have side binding proteins that bind to the side of the actin filaments. Again, we'll see that in the case of skeletal muscles. If we turn our attention back to chapter 11, there we learnt that the inner surface of the plasma membrane needs to be strengthened, otherwise it's too flimsy on its own. And this is where the cytoskeleton comes in its own. The actin filaments form a network to strengthen the membrane region, and that's called the cell cortex. There, actin combines with another protein called spectrin in red blood cells to provide them with their biconcave shape. But however, one needs to remember that in most other cells, they are more packed full of actin filaments than our red blood cells, because those cells have a variety of other functions they have to perform. And the actin in those alternative cells is mostly cross-linked with many other proteins to achieve the alternative functions that they have. Now, rearrangements of the cell cortex can lead to locomotion, as we'll see on this slide. So cells can crawl along the substrate using the rearrangements of their actin filaments. At the leading edge of the cell, the side that's crawling towards the source of something, maybe food, we have the formation of lamellipodium. These lamellipodia are formed when concentrations of actin increase and the actin fibers are pushed forward at their plus ends into the membrane of the cell, forcing the membrane to be pushed forward, as indicated here in the red region. Now, where do these actin fibers come from? Well, they are dismantled at the back end of the cell and recycled to the front end. And as the back end of the cell recycles, it uses myosin motor proteins that can slide along the dismantling actin filaments to assist in the locomotion of the cell. Students need to learn two different processes that can form at the leading edge of a motile cell. The large structures are called lamellipodium, whereas the small finger-like protrusions that you see here and indicated on the subsequent slide, those are called filopodium. The main difference between the two structures, other than size, is what happens internally in the cytosol. In filopodium, actin fibers, actin filaments, are growing in a linear fashion, and they are not branched. Whereas in lamellipodium, the actin filaments are highly branched, as indicated in this slide. And the branching takes place with the assistance of actin-related proteins, indicated in brown, nucleating on the sides of existing actin filaments. So new branches are begun by ARP complexes. And those new branches are stabilized by capping proteins which prevent the further growth of that particular branch. The growing actin network eventually will make contact with the plasma membrane, forcing it further and further out. So this is a real electron micrograph of these filaments. Here is a cartoon of the same thing. And you can see there's a bunch of detail that is missing from the diagram on the right that is present in the actual photograph on the left. Interestingly, integrins, which we'll talk about in the next chapter, these proteins are transmembrane in nature. That means that they are embedded in the membrane itself. So one side of the integrin makes contact with the extracellular world, whereas the other side of the integrin will bind to a growing actin filament, therefore transmitting forces across the plasma membrane into the network of actin filaments. Actin has a large relationship with another protein called myosin. However, myosin exists in several different forms inside animal cells, two of which are discussed in this chapter. So myosin type 1 filaments consist of a single globular head extending into a single tail. And the head region is hinged here, so it can move relative to the tail, utilizing energy from ATP. So class 1 myosin is a very simple molecule compared to class 2, which we'll discuss shortly. Now, the tail region here can attach to various structures inside the cell, including other proteins, and also to organelles. When it comes to organelles, organelles can be moved not just by motor proteins on microfilaments, microtubules, but also by the motions of myosin type 1 along actin filaments. In the first part of the chapter, we were exposed to motor proteins moving along microtubules in both directions. So one thing to notice here is that the orientation of the actin filament is very important in directing motion in one direction only. So this is a one-way motion. If something was to move back along the actin filament, it's unable to do so. So the plus and minus orientation of actin filaments is very, very important. A slight modification of the first diagram is indicated in the bottom diagram here, where the myosin is attached to the plasma membrane. So any motion that takes place along the actin filament here will cause the plasma membrane to move. It may ruffle the plasma membrane, or it may form additional structures. But this is a way in which cells can alter their plasma membrane. What controls the formation of these networks of actin filaments and their associated proteins inside cells? Well, the answer seems to be, in the majority of cases, it's the extracellular environment in which the cell finds itself, whether it's a single-celled organism or a multicellular organism. So cells do respond, as we know from Mr. Gray Plus, and one key finding has been a central protein complex called Rho. Rho is a family of proteins which have a great effect on the cytoskeleton with respect to actin. So in this example here, we have a stain for actin filaments, and you can see the actin filaments have no universal orientation. They're just scattered all over the cytosol of this cell. But in the activation process where Rho is activated, that leads to eventually the alignment of the actin filaments in a particular direction. Other proteins of the Rho class, like RAC, they do something similar in that they can cause various other cellular outcomes. So depending on which protein is activated, the restructuring of the actin network is appropriately organized. So moving on to the next component, which is muscle contraction, we'll find that here actin interacts with a different myosin, and this is known as type 2 myosin. Type 2 myosin is much more complex than the type 1 myosin we just looked at earlier. It consists of two subunits wrapped around each other, and the heads are hinged as well, in that they can move independently of the tail regions, and the tail regions have repeating amino acid motifs along their length so that they repeat various structures. Under normal physiological conditions, the myosin subunits come together to form this bipolar structure, which is quite large. It's 1 micrometer in length. And it's this structure which makes part of the sarcomere that we find in striated muscle cells. So this myosin filament is very important, and it forms naturally in the presence of monomers. Please be reminded that muscle cells, striated muscle cells, are not the only place in which we find association between myosin type 2 and actin. Most cell types other than those muscle cells do contain actin and myosin in various regions. And it's the relationship between the two that allows that particular cell to restructure its shape and to provide internal motion. Returning our attention back to the myosin type 2 filaments, which form naturally in certain cell types, there's a bare region in the center, and that bare region is very important in attaching alternative proteins to the filament. And the other thing to note is that the heads of the subunits, they do not just point north and south, they are arranged in a 360-degree angle so that this filament here could make contact with actin to its left, to its right, to its top, to its bottom, and also diagonally opposed. Let's take a deeper peep inside a skeletal muscle cell. So skeletal muscle cells are also known as striated muscle cells because when you look at these cells under the microscope, you see a pattern of lines like this, striations. And when you magnify that muscle cell, you'll see that those striations have a pattern that can be broken down into something called a sarcomere. A sarcomere is the building unit of a working unit, of a myofibril. And myofibrils are basically bundles of proteins which are found in these multinucleated muscle cells. Students are highly encouraged to learn the information in the next few slides, particularly pertaining to the actions that take place at the physiological level, both with respect to iron concentrations, action potentials, electrical activity, and mechanical force generation, i.e. everything a muscle does. So the actin filaments are indicated in purple, and the myosin type 2 filaments are indicated in green. And you can see the protein of the Z discs is attached only to the actin. The myosin and the actin have some overlap in an extended muscle cell, but upon activation, the myosin will move along both the actin on the left and the actin on the right, causing the sarcomere to condense. So it's important to know that neither one of these proteins actually changes shape. All that's happening is that the two proteins are sliding past each other. It's like interdigitizing your fingers. So if you put your hands together so that the tips of the fingers are touching, and then slide both fingers through each other. That's what's happening in muscle cells of this type. So during contraction, the myosin heads walk along, physically walk along the actin filaments, and during relaxation, the association between the two protein types is reduced to nothing, and the natural counteractivity of other muscles in the body causes the filaments to slide back. So opposing muscles, for instance, your biceps and your triceps, will cause the relaxation of your biceps. At the working level, the interaction between the actin and the myosin is the result of a cyclic sequence of events mediated by the binding of ATP to the myosin hinge region, the head. And hydrolysis of ATP into ADP is an essential step required to detach and then to reposition the head of the myosin molecule. Please be familiar with the deep writing on this page, as it's normally tested upon at a later stage. An earlier question would be, what triggers this event, this sequence of events known as the cross-bridge cycle on the previous slide? And the answer is action potentials arriving at the surface of this particular cell, normally from a motor neuron. So in the human body, nerves that leave various parts of your central nervous system impinge on muscle fibers. And the connection between the two is called a motor synapse. So motor neurons activate bundles of muscles. So when activity is passed down the neuron, it terminates on the surface of the muscle cell. And the muscle cell has a plasma membrane which has evolved to have invaginations which transverse deep into the muscle cells. And these invaginations are called T, or transverse tubules. And their function is to transmit the electrical activity as a wave deep into the interior of these cells so that activation takes place almost simultaneously. Doesn't matter whether you're on the outside or deep inside the cells. Also, we have in green the sarcoplasmic reticulum, another posh name for endoplasmic reticulum. And the plasma membrane and the endoplasmic reticulum are not really touching directly, but there is a special union between the proteins of the two spaces. So in this slide here, we can see the T-tubule, which really belongs to the plasma membrane, and the sarcoplasmic reticulum, the endoplasmic reticulum of these cells, are connected through two types of voltage-gated and mechanically gated calcium channels. So these channels here, they respond to a voltage that travels down the T-tubule, opening them. And as soon as they open, they mechanically cause the alternative types of calcium channels, which are embedded on the surface of the sarcoplasmic reticulum, to mechanically be opened too. So the end result is, almost simultaneously, calcium rushes in from the extracellular space, but more calcium rushes in that's been stored in the sarcoplasmic reticulum. And this is the calcium that's responsible for now interacting in the white space with the sarcomeres. So the structure of the sarcomeres is quite complex. It really involves the movement of three types of proteins. So in red, we have the actin filaments, the ones that are attached to the Z lines. And coating their surface are two other types of protein filament. The first type is tropomyosin, which mechanically blocks the heads of the actin so that the myosin cannot make contact with its binding site. And there's also a troponin protein indicated here that has many subunits that also sits in a groove between the tropomyosin and the actin. The binding of calcium to troponin causes a conformation change which is passed on to tropomyosin. It causes tropomyosin to reposition itself, exposing on the surface of the actin the myosin binding sites. So that's indicated down here. So here's the actin, the same actin that we see up here, and we can see the tropomyosin in purple here. In this instance, it blocks the binding site for myosin. But in the presence of calcium, the tropomyosin is moved by the activity of troponin to a new position, which is indicated right here, and that exposes on the surface of the actin a myosin binding site. So the myosin will be here, and then the myosin will start moving in and out of the page. So please be familiar with this. If you're not sure, check out videos online. Finally, we mustn't forget that smooth muscle cells and cardiac muscle cells also exist inside most vertebrates. The way that nature has come up with different solutions is fascinating. So with smooth muscles, phosphorylation of myosin 2 takes place in the presence of calcium. So calcium in this case doesn't cause a mechanical change, it actually adds phosphate groups to the myosin 2. And that myosin 2 then is reconfigured to act with its target proteins. So this would make the process much slower than a mechanically moved association as we just saw with skeletal muscle. But it is sustained for long periods of time. So we can imagine that your bladder, the sphincter in your bladder, can stay constricted for hours at a time, preventing urine from leaking out. But when it's necessary to urinate, the muscles in the sphincter can be relaxed, therefore leading to a dilation of that particular tube. In addition, because of the way that this phosphorylation can take place, there are a variety of alternative activators that can cause a response. So many cells have receptors for adrenaline, which actually end up releasing calcium, which causes the phosphorylation. We could look at cardiac muscle cells too, but we've decided that that's not really the point of this chapter. Thank you so much.